Hello friends, this video on plant growth and development part 7 is brought to you by examfear.com. No more fear from exam. So now that we spoke about growth, let us talk about differentiation of cells. What do we mean by that? As I explained some time back, some cells will divide to form new cells. Now, only forming new cells is not enough for growth. The cells which are formed, they need to get matured, they need to differentiate, they need to attain a particular shape and size so that they can perform a specific function. And that part is known as differentiation. So it is the process of maturation of cells derived from meristematic tissues to perform specific functions. Now the function of meristematic tissues is to divide, is cell division and formation of new cells. That's it. It will only produce more and more cells. Now those cells need to get matured, they need to perform some specific functions, and they need to attain a shape and size according to the functions which they have to perform. So that entire process is called differentiation. So differentiation always follows cell division. Only this after the cells have been formed, only after the daughter cells have been formed, they will undergo differentiation. So let us take an example. The parenchyma cells, what are they? These are the cells which are formed as a result of cell division. Now these parenchyma cells differentiate. Now when cell division happens, whatever cells are formed, they are all similar. They do not differentiate. Okay, this is parenchyma cell which is formed, or this is sclerenchyma cell which is formed. That is not how it is. So all cells are formed. Now some of them differentiate, they mature in a specific way to form parenchyma cells. Some others differentiate in a different way to form sclerenchyma cells. Now parenchyma cells, their main function is to act as the packing tissue as well as for storage of food. So they have intercellular spaces amongst themselves. So these cells, during the process of differentiation, they need to attain these kind of a shape so that they are able to perform this function. Similarly, if you talk about the sclerenchyma cells, they are mainly for mechanical support. So they should have thick rigid walls. So some daughter cells which were formed, some of them should adapt themselves, or some of them should mature themselves in this way that they attain this kind of a structure like sclerenchyma cells, and that is how they can perform their assigned function. If you talk about the xylem and the phloem, the xylem vessels which you have, tracheids, vessels, or xylem parenchyma, just look at all of them. They are all cells, right? But they do not look similar. If you look at the xylem parenchyma cells, if you compare them with the vessel cells, they look so different, right? Again, when you compare them with the sclerenchyma cells, they all look different. However, they all are cells. They all are formed by the process of cell division. So, process of cell division will just form new cells, that's it. Then the cells need to mature themselves. They need to attain a fixed shape depending upon the specific function which they want to perform. So, this process is known as differentiation. So here on the screen I have given you so many different types of cells, and all of them have different structure, different function, and all these different structure and function comes as a result of differentiation. So this process is differentiation. Now there is another term called de-differentiation. Okay. With the term itself, it looks as if it is going to be more complex than differentiation because you have a D added to differentiation. Okay, let us see what it is. It is a process by which fully differentiated living cells regain the capacity to divide under certain conditions. Okay. Now, sometimes it has been observed that some cells which have differentiated completely, so they are all mature cells now, they are performing a specific function, they have a permanent shape and size of their own. But those cells, suddenly they again gain that capacity to divide. So they again start to divide. So that process is known as de-differentiation. That means the cells which has already differentiated, they are going to produce more cells now. So the process is called a de-differentiation. So on top of differentiation, they are dividing. So divide on top of differentiation. So let us take the most appropriate example for this case would be the parenchyma cells. Now the parenchyma cells which are present, they are fully differentiated cells, right? They have attained their shape, structure, they have intercellular spaces, they are large cells with thin walls, they are acting as storage tissue, they also act as packing tissue. So they are fully differentiated. Now certainly under certain conditions, these cells start dividing again and they form new cells which later form cambium, that is the lateral meristem. I was talking about two types of cambium, right? The vascular cambium and the cork cambium. So they all represent secondary growth. So secondary growth in plant does not happen at the initial stages of their life. It happens only around when they attain maturity. So these parenchyma cells that time again start to divide to form new cells, and those new cells which will later form cambium. So the formation of cambium is an example of de-differentiation, right? So this process happens during the secondary growth of plants. So if you remember the structure of the cambium, this is how it looks like. So here if you see, this is your cork cambium and this is your vascular cambium. So this layer is the vascular cambium. Vascular cambium is formed between xylem and phloem. And this dark colored band is your cork cambium. So these two layers of cells are formed later and they are formed from fully differentiated parenchyma cells. And formation of these two layers results in the secondary growth of plants, which in turn increases the thickness of the plants. Right? So this process is de-differentiation. Now again there is another term called re-differentiation. So what is re-differentiation? Now what is the meaning of whenever you have a re, what does that mean? Just a regain. Regain means to gain again. For example, when you say revisit, that means to visit a place once again. So re-differentiation means to differentiate again. So what happens in this case is, now, in the previous slide, I told you that there are some cells which will de-differentiate to form new cells. For example, the parenchyma cells will de-differentiate to form new cells. What will happen to those new cells? Those new cells will again differentiate. So that means they are actually re-differentiating. So they will differentiate to form the cambium, because they want to perform specific functions. So they will again re-differentiate to form specific functions, like to become the vascular cambium and the cork cambium, and then they will give rise to the secondary xylem and the secondary phloem. Again, the cork cambium will give rise to the cork outside and the parenchyma cells inside. So that process is known as re-differentiation. So the example would be the cambium tissues divide and produce cells which differentiate to perform functions. For example, so here if you look at it, the parenchyma cells, they got fully differentiated, they de-differentiated to form the cambium. That is, new cells were formed which later formed the cambium. What will this cambium do? How will it cause secondary growth? By formation of secondary xylem and secondary phloem, right? So this vascular cambium will later form secondary xylem and secondary phloem. Similarly, these cork cambium will later form the cork, which is also known as the phloem outside, and it will form the phyloderm, that is a layer of cells towards the inside, right? And these are, phyloderm is nothing but the parenchyma cells, and cork is nothing but what we see as bark of the tree. So these cells are formed again. Now this process is known as re-differentiation. So the process by which cells formed from de-differentiated cells divide and differentiate further to perform specific functions. So this is known as re-differentiation. So let us have a quick revision of whatever we have studied so far. So the story started with cell division. Suppose if you have one cell, that cell will divide to form new cells. Now these cells will differentiate to attain a fixed shape and size and perform a specific function. Now once they are fully matured and fully differentiated, sometimes they again gain the capacity to divide again. For example, the parenchyma cells, they get the capacity of dividing again. So they will divide again, and that is called de-differentiation. So for example, the parenchyma cells will divide again to form cambium. Now this cambium will further divide to form cells which will again differentiate to form xylem and phloem, because xylem and phloem again will have different structures. So whatever cells will be newly formed by cambium, some of them will differentiate to form xylem, some of them will differentiate to form phloem. So that process is known as redifferentiation. So I hope the concept of differentiation, dedifferentiation and redifferentiation is clear to you. If not, please rewind the video and see it again. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.